ma'am, and was like one needs to be very positive to such kind of patients. One should like when you're sitting in practice, you should give a positive ray of hope to such patients. Like at Dr. Natarajan's place, we usually give out a handout on what the current research is going on. So the patient doesn't go back with the negative thing. He has something positive to go back to. Like with the modern research now in January 2012, you can see how the research has come up now to humans. So the things are getting better. So one should instill a ray of hope to our patients. Why genes are important? Genes are important because though gene discovery and therapy is not the only path to protecting and restoring vision in people affected by retinal degenerative diseases, it's an essential one. Retinal degeneration affects about 1 in 3,000 of the general population in most countries of the world. As it was said, the retinitis pigmentosa is the most common form, which is one of the most common causes of visual loss in the working years of our lives. RP can be inherited in autosomal dominant, recessive or X-linked manner. X-linked RP can either be recessive affecting primarily only males or dominant affecting both males and females. Some digenic controlled by 2 genes and mitochondrial forms have also been described. DNA testing, as you have seen earlier, is available for many types of RP. Genetic mapping and identification has been done for X-linked RP, the most devastating form of RP. RPGR gene by positional cloning was identified and later novel isoforms of these gene which contains a mutational hotspot were identified. Mutations in RPGR gene account for the majority of X-linked RP patients and an estimated 20% of all RP patients. RPGR is a novel protein and current efforts are aimed at elucidating its function in health and disease. RPGR-P1, now known to cause LCA as a form of congenital blindness. This protein appears to anchor to the connecting cilium of photoreceptors, implicating RPGR in the polarized transport of proteins to the outer segments of photoreceptors. And now coming to macular dystrophy. Macular dystrophy is a heterogeneous group of disorders characterized by progressive central visual loss and degeneration of the macula and underlying RP of the eye. These disorders can reveal visible fundoscopic changes, physiological changes in visual diagnostic tests of both. Genes for inherited macular dystrophy such as Stargardt's and BEST have recently been mapped to specific chromosomes. Now the classification of inherited macular dystrophies are either primary hereditary macular dystrophies or rare hereditary macular dystrophies and macular abnormalities with generalized chorioretinal dystrophies. Now coming to Stargardt's macular dystrophy, originally described by Stargardt in 1909, is the most common hereditary macular dystrophy and accounts for 7% of all retinal dystrophies. Typically, patients presenting with decreased central vision, usually in the first or second decade of life. Fundoscopically, it is characterized by bilateral atrophic changes in the macula, degeneration of the underlying RP, and the presence of prominent yellow-white flecks in the posterior pole. Although Stargardt macular dystrophy was originally described as an autosomal recessive trait, several large families have recently been reported with an autosomal dominant mode of inheritance. Recently, a gene for recessive Stargardt was mapped in the short chromosome of 1 in 8 families. This result is consistent with genetic homogeneity and suggests that possibly only one gene is involved in this form of disorder. In contrast, several genes have been implicated in autosomal dominant form of the disease. Of families studied so far, 2 disease genes have been localized to the long arm of chromosome 13 and long arm of chromosome 6 respectively. As more families are studied through genetic linkage analysis, more genes may be discovered in the future. Researchers are working on gene therapy for treating Stargardt's. In a mouse model, they are working on efficiently and effectively inserting a healthy version of ABCA4 gene into the photoreceptor cells. Ultimately, the researchers hope to insert healthy version of the gene into the retina of patients with Stargardt's disease. BEST macular dystrophy. This disease is an autosomal dominant inherited disorder first described by Dr. BEST in 1905. Patients are found to have bilateral macular lesions at a very young age. Typical fundamental findings include a yellow egg yolk-like appearance of the macula. Visual acuity usually remains fairly good for the first 5 decades of life, but eventually deteriorates in the 6th or 7th decade to legal blindness. A 5-generation family with 29 effective members has been studied. Linkage analysis mapped the disease-causing gene to chromosome 11, Q13. Subsequent studies of additional families indicate the gene in 11q13 is a major, if not the only gene responsible for BEST disease. RUM, which is specifically expressed in the outer segment of photoreceptor cells, appears to be a promising candidate gene for this disorder. However, no definite mutation with this gene has been found in any patient to date. Gene replacement. Extensive efforts have been made over the past 15 years to develop ways to deliver normal genes to the photoreceptor and RP cells. Carriers known as vectors are required for delivery of therapeutic gene to the target cell. Adeno-associated viral vectors. Viruses are the most common vectors for gene delivery. Viruses have evolved a way of encapsulating and delivering their genes to human cells. Scientists have manipulated viruses to remove disease-causing genes and insert therapeutic ones. Advantages of such vectors is that safe, non-toxic, no significant human immune reaction, long-lasting, single injection is needed, widely used in gene therapy studies, and the disadvantage being it's used mostly for small genes. Ribozymes. Some RNA molecules termed as ribozymes have been shown to have catalytic activity. Hairpin and hammerhead ribosomes are being used as tools for gene therapy. Ribosomes are being designed to block retinal degeneration in certain forms of RP. These have been tested in animal models of this dominant genetic disorder using AAV vectors. This approach has been extended to combat diseases affecting the brain, Huntington disease, peripheral nervous system and heart, familial hypertrophic cardiomyopathy. Widespread application of linkage analysis and mutation detection techniques, complex underlying pathology has now been revealed, and several lines of research raise the hope of genetic treatment. So one should not lose hope at the end of the day, and you should give hope to your patients of genetic diseases. I would say that this is the concluding thing one should take back home message from the IC.